All right. Well, hello, everyone. Welcome to today's session of our CPH review session series from the National Board of Public Health Examiners. My name is Ashley Mueller, and I'm the Manager of Educational Programs at NBPHE. As in the last few sessions, I'll continue to monitor chat and Q&A throughout the presentation so you can interact with your audience members. And if you have questions for our presenter, please make sure to use the Q&A so that we see that and the presenter can answer the questions at the end of the session today. As we have been doing the last few sessions, we will be raffling off another copy of the APHA CPH exam review guide. So anyone who's here live, you are automatically entered in that raffle and you have a chance to win that guide. So now on to our topic. We are going over public health biology and disease risk today, presented by Dharshini Yogendra. Dharshini is a board-certified pediatrician and also certified in public health. She has worked in the public health sector for the last 10 years and currently works as a school physician / medical city medical specialist for New York City. Her specific public health interests include school health, immigration health, and preventative medicine. So with that, I'm going to turn it over to Dr. Yogendra.
Hi, everybody. Good afternoon. Thank you so much for having me. I'm really excited to be here. Thank you for the introduction also, Ashley. Like Ashley just mentioned, I'm a pediatrician. I'm a board-certified pediatrician. I work for the New York City Department of Health and Mental Hygiene as a school physician, and I'm here representing myself and not my employer. I do have to say that. I am really excited to be here, and I'm excited to be here to be able to talk to you about this incredibly interesting aspect of public health, which is public health biology and disease risk. Okay, disease risk, and it's a pretty big topic. We do have a lot to cover, so I'm going to move pretty quickly. And we do have a lot of slides in this presentation, and I have included some charts that I will make sure get to you at the end of our webinar. And I hope that you find this useful in your exam prep. Okay. I know you've been seeing this many times in the last few weeks, just one more time to see it, just a breakdown of what the exam is. 10% of the exam, like every other section, is with public health biology and human disease risk. Okay, so this is our roadmap for today, how we're going to do our talk, and these are the key topics we're going to cover. So first of all, what is public health biology? I mean, we hear it a lot. I think intuitively we know what it is. The way we can think of public health biology is by being the biological and molecular context of public health and its application to public health practice. So basically, in very, very short terms, it's a biological perspective on public health issues. The most important tenets, or one of the most important tenets of public health, is understanding causation, right? Why does something happen? Why does this illness happen? Why did this event happen? We've come a long way in understanding diseases and why disease happens in the last 100 years or so. And to understand how we got to this point, I thought it would be useful to just take a quick trip down memory lane to see how the road we've traveled on in public health history. And public health history is absolutely fascinating. So if you ever get the time, it's good to just flip through and read through the history. Some really, really cool stories. I don't want to take too much time that we have together to talk about this, but just to highlight some stuff. In the really early times, diseases occurrence were explained by superstition, religion, myths. It was often thought that disease was a result of sin. So practices were done to treat illnesses to reflect this thought process. But of course, there's no systematic or evidence-based way to have studied this idea. The bubonic plague hit us in about 1300s to the 1700s, and it was a series of epidemics that just ransacked Europe and Asia with thousands of people dying. And at the time, the concept that was the dominant theory as to why the disease was happening was called miasma theory. Miasma theory believed that disease happens because of poisonous air. So if you look at this picture here, this is a plague doctor. So it was a doctor covered from head to toe with this beak, of course. And in that beak was actually filled with aromatic substances that cleanse the air. And this is, in a way, is sort of the first hazmat suit, if you think about it. And they also use ideas such as quarantine and isolation. So similar things that we're using now. But even still, they had this theory of miasma that really couldn't really quite be studied. The causation couldn't really be studied. It was more like speculation. The Industrial Revolution came along afterwards, and there was a burst of economy with a mass migration and a massive growth in population and city growth. And with this brought a myriad of public health issues that we had never seen before. Cramped housing, poor work conditions, poor ventilation, really, really serious sanitary issues as well. So the need to improve public health practices was higher than ever before. So around 1800s and 1900s, cholera happened. And if you can recall from your studies, or you may have heard of the work of John Snow, who was a British scientist who in 1855 was able to systematically test and prove his hypothesis that waterborne diseases actually cause cholera and not miasma. And his breakthrough work was one of the first major studies that challenged the theory of miasma in a very evidence-based way. Germ theory had always kind of floated around, the idea that germs cause diseases. Scientists had always dabbled with it, with the idea, but Louis Pasteur in 1870 was the one who really kind of articulated it in a really great way when he studied silkworms, because the silkworms were dying and the French industry of silk was suffering. So they recruited him to study why this was occurring. And he noticed that he understood that there's a role of microbes in disease and disease spread. That was further expanded on with Koch's postulate in 1890, which established criteria to demonstrate the relationship between organism and disease. And you can read about it over here. What the postulates really say is that a microorganism must be found in abundance in animals suffering from disease, and it should also not be found in a similar animal that's healthy. Then you can isolate the microorganism and grow it in a culture, and that cultured microorganism should cause a disease when it's introduced into a healthy organism. And then you could take the microorganism out of the inoculated animal and see that same exact microorganism there. So really, it's taking a healthy animal and a sick animal and comparing the two to prove that disease is caused from a microorganism. Of course, this is the most basic way of thinking of disease spread, and we've come a long way since then. So we now know that it's a little bit more complicated than that. It can be a little bit more complicated than that regarding disease spread. But that was sort of the first idea of germ theory that was really articulated to the public. Louis Pasteur went on to use heat and pasteurization to develop vaccines, and that opened a whole new many, many years of lots of vaccine development. And then finally, we got to the point where we are now, which is genetics and understanding genetics and the genetics role in disease and illnesses. And now we're doing groundbreaking work with what we know about genes, such as gene therapy and using genetic-based vaccines as well, which we're going to talk about later on. So that's a very, very brief and quick history of public health and understanding causation. So this all takes us to the basic of us understanding public health biology. What we have learned through evolution of public health is that diseases and events are not random. They occur because of a mix of our biology and our environmental influences. And largely, they occur because of the presence of risk factors from genetic risk factors as well as environmental risk factors. And what are these risk factors? How would you define risk factors? Risk factors are characteristics or exposures that increase the likelihood of developing a disease or illness. For example, smoking, the habit of smoking is a risk factor for developing lung disease. Risk factors themselves are not random, but they are influenced by biological and social determinants of health. So to refresh your memory, what are social determinants of health? And I know this has been discussed in other presentations. Social determinants of health, to remind you, are conditions in the environment where people live that affect their health, functioning, quality of life, outcomes, and risks. So things like healthcare access, educational access, economic factors, built environment. To understand what risk factors may be playing a role in the development of disease, it's useful to look at patterns of spread, like certain areas of the map. If you can look at that and see that there is a kind of a cluster of diseases, that can help us kind of backtrack and look for risk factors as well. A question to ask is, how does a risk factor differ from a hazard? Sometimes they're used interchangeably, but they're not the same thing. A hazard is more of the characteristic of an agent that can cause harm. And a risk factor increases the probability of an adverse effect occurring when exposed to a hazard. For example, pesticides, right? We can take pesticides. Pesticides such as organophosphates can be considered a hazard, as exposure to organophosphates can increase your chances of getting poisoning or even death. Risk factors would be considered your work occupation or like your work conditions. That's a risk factor for being exposed to organophosphates and getting organophosphate toxicity. So I hope that makes sense. Here's another example to help us think about risk factors. And we can see here that there are a myriad of possible factors that play a role in the development of uterine fibers. And that can include physical activity, diet, hormones, age, race, genetics, and endocrine disruptors and lifestyle. So risk factors related to the development of this illness. A way that we can understand causation is by using models of disease causation. So models of disease causation help us understand disease transmission. And this is the most well-known one, the epidemiological triangle. This triad helps us understand the relationship that exists between agent, environment, and host. And we can see that very clearly here. An agent is anything that causes diseases. It can be biological, like a bacteria or a virus, or it can be chemical or physical. And it interacts with the host in a specific environment. And each component of the triad also has specific characteristics that play a role in how the interaction will be. And you can see these kind of possible characteristics in the corresponding boxes. For example, the environment could be warm, which may be a thriving condition that encourages the proliferation of microorganisms. The agent can have resistance to antibiotics, or the host can have a high risk, can be at an age that makes them high risk. So that makes them more susceptible to the toxicity of the agent. And by understanding this model, the characteristics of the components, as well as the process, we can also work towards figuring out how and where we should focus interventions to mitigate disease. Let's look at an example. We can take Legionnaire's disease as an example of how to apply this model of disease causation. Legionnaire's disease is caused by the bacteria Legionella, and Legionella is generally found in natural environments and natural freshwater sources like lakes and streams. But it becomes a problem when it gets into a man-made water system, like cooling towers. Cooling towers use water to cool air and air conditioning units, hot tubs, water tanks, and they get in there, and it's a great environment to grow and thrive. The temperature is great for them. And they like to grow at a temperature of 77 to 113 degrees Fahrenheit. It is then aerosolized and inhaled, getting into the lungs of susceptible individuals. So the people that are most susceptible for this are people who are over the age of 50, former or current smokers, or those with compromised immune systems. In this example, we can understand key characteristics from each component: the agent, how the agent works, the host, and the environment that allows for the Legionnaire's disease to occur. And also, by understanding how transmission works here, we can also think about intervention strategies that can be utilized and what step of the interaction these strategies may be implemented. For example, we know that we can figure out where we need to clean the tubs, or where we need to check the pH and provide education. If we knew that natural environments, Legionella traveling from natural environments into water systems are what's causing a problem, we can figure out a water management program to mitigate that as well. Okay, so talking about Legionnaire's disease and the agent-host environment interaction leads us to our next question, which is, how are infectious agents transmitted? And this diagram, the chain of infection diagram, is a useful way of thinking about how this happens. We start right over here with the causative agent, which is a pathogen, bacteria, virus, fungus, parasite. And they like to make themselves at home in a place called a reservoir. And a reservoir could be anything. It could be soil, food, water. It could even be a person. An example of this would be the herpes virus. The herpes virus lives in the dorsal root ganglia. It's a reservoir for the virus. The agent then leaves its home through a portal of exit. And the portal of exit can be multiple different ways. Sticking with the herpes virus, for example, the virus leaves through shared secretions or contact of mucous membranes. The agent leaves and then travels to a host. There, at various forms of travel, direct contact, indirect contact, or through vectors. And we're going to talk about this more in the next couple slides. In our example with herpes, herpes most commonly gets to the host through direct contact, usually mucus to mucus contact or saliva to mucus contact. Once it gets to the host, it needs to figure out a way to get into the host via a port entry, which should be through the mouth, the nose, eyes, cuts in the skin. And then we think about how some hosts might be susceptible, right? Elderly people, infants, immunocompromised. or really anybody. Sometimes there's really no susceptibility people, everybody's sort of at risk as well. So this is the frame of how infection occurs and how an agent can infect a host. But we can use this to also think about the chain of transmission when we think about disease prevention and even with eradication to figure out mitigation strategies and management strategies, public health strategies. When we talk about modes of transmission, there are various ways agents get to their hosts. And remember, some agents can use multiple modes of transmission. One is via direct contact, so direct contact with fluids, tissues, secretions. And direct contact examples are person-to-person spread, like STDs. Some perinatal infections that occur during delivery, such as herpes simplex or group B strep or Ebola. Another is via indirect contact through transmissions through a contaminated object. And influenza and norovirus are two very popular viruses that can be spread through a contaminated object. If you recall, sometimes if you're in the cleaning section of the grocery store, they have wipes that, they have like cleaning wipes and they'll say 99.9% of viruses or bacteria eliminated. That's what they're aiming for, these kind of viruses that live in these bacteria and viruses that live in these kind of contaminated object areas. Other modes of transmission, airborne transmission. And so when pathogens can be transmitted through the air, like via small droplets, inhalation, skin, GI, eyes, hearing, things like SARS and tuberculosis and varicella. And this is when it's just coughed on the air and it just kind of floats around in the air. That's really what airborne transmission is. Oral transmission is when pathogens are transmitted when contaminated substances or foods are consumed. We can see this, for example, with cholera and listeria. And vehicle transmission is when a non-living object carries an agent. So an example of this would be fomites. So an inanimate contaminated object is carrying the agent. And a vector transmission is really important that we talk about a lot in public health, which is when living organisms transmit infectious agents between humans or between human and animals. And with vector transmission, insects are the most common vectors. And we're going to talk about this more now. There are two types of vector transmission, mechanical and biological. With mechanical, it's pretty straightforward. The vector picks up an infectious agent on the outside of its body, and then it transmits it when it lands on a new host. So a good example of this are the houseflies. Like a fly will land, say, for example, on cow dung. It gets its legs dirty in cow dung, and then it goes and lands in food, and then someone comes and eats it, right? That's pretty A to B, pretty straightforward. The more complicated and the one that's associated with more devastating diseases is biological vector transmission. And this is when the pathogen actually infects and grows within the vector. So it needs the vector to stay alive. And it harbors the pathogens which grow inside of the vector. And the vector in turn injects it into a host. And diseases that are usually associated with this are things like malaria, Lyme disease, and Chagas disease as well. And this is a picture right here of how it really works. A mosquito bites a person who becomes infected, infects the liver, and it's just a cycle that goes on and on. Here is a list of frequent, common, or ones that we hear a lot of diseases associated with biological insect vectors. You can see them listed over here, as well as the causative agents and the type of vectors, well, mosquitoes. And you can see mosquitoes play a huge role in vector-related diseases internationally around the world. It's a huge problem. And there are a lot of mitigation strategies that are used to deal with these issues. I think for the exam purposes, I wouldn't focus so much on learning the names of the specific vectors, but more focus on, a mosquito versus a fly, a flea, what exactly the vector type is that's associated with the disease and the causative agent. I think that would be probably a more high yield thing to focus on. And right over here is a picture of a very, very common clinical sign we see in Lyme disease, which is that target rash associated. When we see that, we usually kind of think almost right away, it's probably Lyme disease, or it could be Lyme disease. So question, this is just for us to kind of talk through on our own. What is the chain of infection a model of? So
yeah, you can put that in the chat. correct that we didn't put a poll together for this one,
Yeah,
right?
yeah,
Okay,
thank you.
yeah. So why don't you all put your answers in the chat?
Okay, good. And most everyone is saying the same thing, which is awesome. D, the chain of infection is how pathogenic genetic microorganisms are transmitted from one person to another. That's exactly right. Obviously, it's not about how bacteria multiply, and it's not about how it affects the immune system. The other one you can kind of think about maybe is how infections can be prevented, which is, if you think about it a little bit, maybe it's not the best answer, but we can use understanding that model to understand where we can implement strategies. But the model itself is a model to understand how microorganisms are transmitted. So great job, everybody. All right, close this out. And that will take us on to our next section. And okay, I'm doing good with time because I practiced this and I went over a bunch of times. So I'm timing myself pretty strictly. Anyways, okay, so we talked about disease causation. Now let's delve into how we keep an eye out on the development and spread of diseases and practices to reduce spread. And this is an extremely important part of what we do in public health biology. Disease surveillance means the systematic collection, analysis, and interpretation of health-related data. So basically getting information on how a disease is affecting population. Surveillance is important because it helps monitor disease. It basically tells us what the disease is doing, so we can figure out what we need to do. So it helps monitor disease trends, describe the nature, the natural history of the disease, identify any epidemics, new syndromes. Maybe helps us see that infectious agents are maybe changing, like they're not responding so well to treatments that has worked in the past. It helps us identify areas of research and also helps for policy development as well. So surveillance is very, very key in what we do. And there's several methods of disease surveillance, which you can see here in this chart. And I organized this all so that you can kind of compare it all easily. There are three common types that we use: passive surveillance, active surveillance, and sentinel surveillance. Passive surveillance is the one that's most commonly used. So that's basically the health agencies or healthcare facilities they passively send information over to healthcare agencies, to public health agencies, sorry. They kind of do it on their own. It's pretty inexpensive, but it's also questionable quality of the data because like we're leaving it up to the healthcare facilities to just send out information, which may or may not happen, or they might not properly report things, or they might forget to report things like that might happen. Active surveillance is when data is requested by the public health agencies from healthcare facilities. And this is like more accurate data collection because they're specifically asking for things, but it does take more time and more resources. And similarly, with sentinel surveillance, data is requested from a small group of healthcare facilities. And this is very useful because you can obtain more detailed information on specific illnesses. But this also takes more time and resources as well. And I will make sure that these charts are given that I'll get these charts off to you because I created them for study purposes. So I hope that you will find them useful during exam prep. I mean, when I study for exams, I always like to organize information in charts. So hopefully you'll find this useful. And when we talk about prevention of diseases and spread and outbreak, there are many different types of public health practices that we can do this. Pasteurization is one of them. We talked about it a little bit before. That's when heat is applied to food, even with vaccines to kill off pathogens. Disinfection, hand washing, sanitizing, and things like that. And actually, improved sanitation and hygiene has had the greatest impact on lengthening life expectancies. So this is probably the biggest thing that we did in the last couple hundred years in terms of public health practices to improve life expectancy. Barrier contraceptive methods such as condoms used to protect STDs, antibiotic use to eradicate bacteria, quarantining, and vaccinations. And now we will talk more about vaccinations. So how do immunizations work? So vaccines are a big part of what I do as a pediatrician. So I am very excited to talk about this. But immunizations basically are self-defense trainers. That's how I think about them. They teach the body's immune system how to recognize and fight disease-causing agents. It's an example of active immunity, which is when the body is triggered to make its own response. And we're going to talk about this more in the next couple of slides. And this is just a very basic explanation of how vaccines work. Here is another chart with 6 different types of vaccines that are available for us today, as well as examples. Inactivated vaccine uses the killed version of the pathogen in the vaccine, and that allows you to mount immune response against the killed version. These vaccines sometimes need boosters. Examples are Hep A, influenza, and polio. Live attenuated vaccines use a weakened form of the pathogen. So the pathogen isn't completely killed off, but it's very weak. And they tend to have strong lasting effects, but a caveat for immunocompromised individuals, they might be able to mount that strong enough immune response. So they might actually, get the disease. So we always are very, very careful using live attenuated vaccines. In immunocompromised generally, we don't. And examples of this are MMR, varicella, and rotavirus. Another group is subunit, recombinant, polysaccharide, and conjugate vaccines. This is when specific parts of the pathogen, like the protein, the sugar, the capsid, are used. The examples are listed there. Toxoid vaccines use toxins created by the pathogen, and the immunity is created against the toxin and not the pathogen. And sometimes you do need vaccines as well. Examples are diphtheria and tetanus. That's why you need to get your Tdap shot every 10 years. Viral vectors and mRNA vaccines, we've been hearing a lot about in the news because both of them were used during the COVID-19 pandemic. Viral vectors use a harmless virus that carry a modified version of genetic info of the pathogen, and mRNA vaccines use a pathogen's RNA to make specific proteins to trigger an immune response. So these are the 6 major types of vaccines that are available out there. And just to talk a little bit about immunity, just immunity 101. Active immunity is when the body is triggered to create antibodies. Your body is being triggered to make antibodies. So this can come from like vaccines, because your vaccines will trigger your body to do that, or if you get the disease itself, because then you have to mount a response to fight the microorganism. It can take some time to build up, and it can be long lasting. Passive immunity is when you are receiving antibodies in one way or another. Infants can receive antibodies through breast milk. Fetuses can receive antibodies through placenta, or you can receive antibody-containing products such as immunoglobulins, which we use in conditions like Kawasaki, certain leukemias as well. These effects are usually acute and immediate. We can also think about immunity in a broader scale, like innate versus adaptive. Innate immunity is the first line of defense that we're born with, whereas adaptive immunity is what we acquire, like getting vaccinations. It's what we acquire during our lifetime. And third, herd immunity is when many people have immunity to disease, making it harder to spread the disease in the community. It's especially important because it's a protection for those who can't get the vaccine, either because of their age or their medical condition, or because of an immunocompromised status. So this is a nice picture kind of showing how the strength of herd immunity. In that top picture, most of those people are healthy. The blue people are healthy, but not immunized. And you can see a couple sick people in that group can make everybody really, really sick, right? As opposed to the second picture, the below, which is the people that are in yellow are immunized and they're healthy. And they are able to protect one another more because there's less likely spread, since most people are immune. They're not going to get the disease. They're not going to be symptomatic. And so people are protected. But keep in mind, between 80 to 85% of the population must be immunized to prevent the spread of disease. And it's even higher for really, really infectious airborne diseases, such as measles. Okay, question for the group. New vaccines for influenza must be developed every year because, and we didn't quite talk about this, but hopefully you know about this. Okay. Yes, very good. Most everyone is saying A, which is the right answer. New strains of the virus evolved that are not affected by existing viruses. And that is the correct answer. None of the other ones really make any sense at all. But influenza viruses mutate frequently, producing strains for which the body doesn't have antibodies. And that's why we have to get a new vaccine every year. And that is called antigenic drift. So great job, everyone. Okay. Moving on to non-communicable public health issues. All right. Injury and prevention. This is a huge public health burden at this point in the world. Injuries are the leading cause of death for individuals ages 1 through 44. And previously, motor vehicle accidents were the #1 cause of injury and violence and deaths. And it was for a very long time. But now, its #1 cause of death is unintentional poisonings. And this is because of the rampant use of opioids. People are overdosing, unfortunately. And you can see the change in numbers, that this poisoning is just getting bigger and bigger. And that's all due to the opioid crisis. Let's switch gears a bit and talk a little bit about the non-communicable diseases themselves, such as chronic diseases. And they are the leading causes of death globally. They kill about 41 million people worldwide yearly, exceeding the deaths from communicable diseases. Commonly, cardiovascular disease, cancer, chronic respiratory disease, and diabetes as well. And there's a reason we switched from communicable diseases over to non-communicable diseases in terms of the burden of public health in developed countries. And that's because we've done so well with understanding public health and improving our public health practices and our medical advancements. And that shift towards non-communicable diseases is epidemiologic transition. That's what it's called. There are various factors that play a role also into why non-communicable diseases are predominant. A big one is aging population. People are living longer because we've done so well with public health practices and medical advances. They have increased life expectancy. The median age has increased as well. And we now have advances, like I said, in disease and diagnosis management as well. We have improved access to care, antibiotics, improved sanitation, improved understanding of genetics. So people are just living longer. And so they're having more time as they live longer to see chronic diseases develop. So talking about genetics, let's talk about genetic screening, because we have this available, which wasn't available hundreds of years ago, but we have the ability to screen for genetic conditions prior to a baby being born. We have things such as prenatal screening available. We have quad testing, amniocentesis, chorionic villus sampling as well. And these tests they check for things like trisomy 18, trisomy 21, and neural tube defects like spina bifida. And they help make decisions. Having this information beforehand, you'll know when someone needs to be a little bit more careful during pregnancy, things like that as well. We also have another screening test available called the newborn screening. And newborn screening is done in every state. And it's a blood testing done after 24 hours of a baby's life that checks for a multitude of disorders that are a combination of genetic, metabolic, blood, or hormone. Just lots and lots of diseases that are tested very early on. It's required by every state, but each state tests have different specific tests that they're testing for. Generally, there's a lot of similarity because there's some major ones that we test for, such as PKU, sickle cell disease, congenital hypothyroidism, galactosemia, congenital adrenal hyperplasia, and cystic fibrosis. And most of the diseases that are tested for follow an autosomal recessive pattern of inheritance. And this is really great because now we're able to pick up conditions earlier on in a baby's life that we can treat very, very early on. Some of the things that, if we wait longer to make a diagnosis, they can have long-term repercussions, such as things like congenital hypothyroidism. It's one of those conditions you just need to start treatment right away. You don't want to wait because it can affect the intellectual abilities of a baby, and that will be long-lasting. Congenital abnormalities. So we can talk about congenital abnormalities in different ways. Chromosomal disorders are when there's a change in the number of structure of chromosomes. So like Down syndrome, which is trisomy 21, trisomy 18 is Edwards syndrome, Turner syndrome is XO. These are examples of when there's a different number of chromosomes than what we usually see. Single gene defects also occur. And what this is when there is a defect in one specific gene is what causes a disorder to occur. An example is cystic fibrosis, where the gene defect is on chromosome 7, sickle cell disease when the defect's on chromosome 11, and PKU when the defect is on chromosome 12. And teratogens. Teratogens are agents that induce a structural abnormality, growth deficiency, or functional alteration during prenatal development. And you can see over here, I took this from Up to Date. This is basically a breakdown of organ development during a fetal life. The first organ to start to develop is the CNS. It's as early as the end of the second week. And those black bars that you see here are the times that these organs are at higher risk for the effects of a teratogen. And as you can see, most of the risk is associated very, very early on in the pregnancy, kind of in the first 2 months, really, the 2 to 3 months. So, and that's important to keep in mind, because sometimes people don't even know that they're pregnant for a few months and may unknowingly be exposed to teratogens. So it's important to know that the earlier part of the pregnancy, earlier gestational weeks, are the time when an infant and fetus is a high risk for congenital abnormalities due to the influence of a teratogen. Common teratogens, infections are a big one when a mom gets infections. Torch infections it's an abbreviation that we use. Toxoplasmosis, others such as Zika, syphilis, parvovirus, rubella, CMV, and herpes. And they present in different ways, but as a whole, a lot of them come with microcephaly, heart problems, limb problems, chorioretinitis, jaundice, low birth weight. There's a whole slew of different kinds of ways they can present. I wouldn't focus too much on knowing the specific presentation of each. I think the one that you really should know is probably toxoplasmosis. Toxo is a parasite that you can get by eating uncooked or undercooked meats and shellfish, drinking contaminated water, or from cat feces. And for most people, getting toxo it's a pretty mild disease. But if you are pregnant and you get toxo, you have a high risk of passing it on to your fetus, and the fetus can have effects of that. Things such as microcalcifications, as well as microcephaly and calcifications in the CNS system, which can cause neurological problems as well. So that's why it's important to take caution with cats and dealing with cat litter when one is pregnant. Other common teratogens, maternal diabetes, can be associated with congenital heart disease, cleft palate, neural tube defects, and bone issues as well. Physical agents can also cause issues. Heat, if a mother is exposed to heat for prolonged periods of time, that can have some effect on the growth and development as well of the fetus. And ionizing radiation can also cause growth disturbances and issues as well. And that's why, of course, pregnant women are not supposed to get X-rays. Drugs can also play a role. Alcohol, tobacco, oral isoretinoin, anti-seizures, and thalidomide. So alcohol is not safe at all during any time of the pregnancy. There's no particular time that they say that is safer than the other. They just the entire time, it's just not safe for a mother to consume alcohol. And babies that are exposed to high levels of alcohol can have neurobehavioral disorders, birth defects, neurocognitive deficits as well. And this picture right here is the clinical manifestations that we see sometimes in children who are born with fetal alcohol syndrome. Oral isoretinoin is also known as Accutane, and that can cause issues with ear anomalies, CNS malformations, lots of defects associated with that. And very, very well known is thalidomide. Thalidomide was a drug that was commonly used in the 1950s and the 60s for morning sickness and in pregnancy, and also as a sedative. But it was found to be the cause of thousands of miscarriages and over 10,000 children born with deformities. Many children develop this thing called phocomelia, which is when their extremities look like flippers. And these are just pictures over here that you can see. These are of babies that were born to moms who took thalidomide. Smoking is associated with poor fetal growth as well, because it has an effect on the placenta and the vasculature as well. And chemicals such as lead can lead to neurobehavioral disorders, and mercury exposure can lead to CNS damage as well. All right, so this takes us on to our next big section, which is hazard exposure in the physical environment. And this is a big section, but it's really, really interesting. So I'm excited to talk to you about this. Public health biology of our physical environment. Our physical environment includes air, water, food, soil, and our built environment. And our built environment includes the things around us, like our means of transportation, housing, and development. And of course, our health is linked to all of these. So we're going to talk about them one by one, and I promise you it is interesting. Food safety. Food becomes contaminated with an agent due to several possible reasons, and I'm sure we can think of a few just off the top of our head. Obviously, poor hand washing during food preparation and serving, cross-contamination with other contaminated foods, inappropriate storage and cooking temperatures, or contamination by animal waste. The temperature range, which is risky for allowing the growth of microorganisms and could lead to food illness, is between 40 to 140 degrees. So that's the temperature that the microorganisms will thrive in, 40 to 140 degrees. Remember that. That is important. And foodborne illnesses occur when this contaminated food or beverage is consumed. Ensuring food safety is kind of a joint effort of the FDA, the USDA, and the EPA. Local and state health departments have the primary responsibility of monitoring safety and gathering info on outbreaks, and they in turn report it to the CDC, and the FDA gets involved if it's FDA-regulated substances. I was just looking at this the other day, and I was making like a veggie burger, and they said to cook it, make sure the internal temperature is 165, and that always stayed in my head. And I'm looking at this now, and I see that this is like right up here. So I just remember that 165. Okay. So when we talk about infectious food hazards, we can kind of think about how to categorize infectious agents in our head using this graph. This graph is diagram is really useful, I think. Because we want to think about what organisms are associated with what food. And some organisms can be in multiple types of foods, like Salmonella and Listeria can be in raw vegetables as well as in poultry, as you can see here. But this is a very nice way to organize that information. A focus on Listeria, right? Listeria in pregnant women can be very risky to both the woman and the fetus. And Listeria can be in soft cheeses, unpasteurized milk, as well as in raw foods as well in poultry. So always important to keep that in mind. The next couple slides I put together just to show what the organisms and the associated food in the presentation are. The presentation is like very, very similar for all of them. There are sometimes only mild differences to help you differentiate what the illnesses are, always usually with stomach cramping and some form of diarrhea. I won't go into this in too much detail. Again, I'll make sure that you get these charts for your exam preparation. But just to highlight, Bacillus cereus oftentimes associated with rice. Botulinum is associated with canned foods, and less than 1 year of age, it's in honey. So that's why we don't recommend children under 1 year of age to eat honey, because it can have botulinum spores. And adult GI systems can break down the spores, but infant GI systems are not developed enough to break down the spores. So that's why we say avoid honey in kids less than 1. Campylobacter raw undercooked poultry, and it is the #1 cause of bacterial diarrhea in the US. With E. coli, water, food contaminated with human feces. It's also known as traveler's diarrhea. There's a subset of E. coli as well, which you don't need to know, but E. coli as a whole. E. coli 0157 is important to know because it's associated, you can get quite sick from it. It's associated with severe and bloody diarrhea and a risk of developing hemolytic uremic syndrome in kids. And that can eventually lead to kidney failure also. So this is very important to know. It causes vessel disease and can cause a lot of bleeding and kidney failure in kids. I mentioned Listeria, unpasteurized milk, soft cheeses, deli meats as well. Norovirus it used to be associated with cruise ships. I don't think it's super common, but for some reason it's always associated with being on cruise ships. That's how I remember it. Salmonella, eggs, poultry, meat are a big one, as well as contaminated veggies. Shigella is raw produce. Vibrio is cholera. It's associated with undercooked raw seafood as well as contaminated water. And Hep A is with raw produce as well. You can have flu-like symptoms, diarrhea, dark urine, and jaundice. But now we have a Hep A vaccine available that we use pretty routinely in our kids. So hopefully people will start to get protected from that more. The hazard analysis critical point management system is a tool that we use, and this helps us prevent foodborne illnesses before it happens. It's used to identify, evaluate, and control food safety hazards so foodborne illnesses can be prevented. And the goal of the system, again, is to evaluate every step of the food process to reduce risk. So it's a prevention strategy. And there's 7 steps that you can see listed here. The first step is doing a hazard analysis. So consider the possible food hazards that can occur at every aspect of the food process and determine which hazard has the highest risk. And the next step is very important. And if you're not going to remember anything from this slide at all, because I know there's a lot, just remember this one step, which is understanding and identifying critical control points. Critical control points are key points in the entire food preparation process where a hazard can be prevented eliminated or reduced to a safe level. Right. So it's where we know, it's the points that we know that we can, that the hazard can be prevented. That's the most important part of this whole slide and knowing this whole process. But also the next step is to determine critical control points. So the next step is to set critical limits and then to create a monitoring system, establish corrective actions, what must be done if the guidelines aren't being followed. You need to periodically review guidelines. And step 7 is to make sure you keep record of everything that we're doing as well. And that will help understand the system a little bit more. So again, the hazard analysis critical control point management system is used to help evaluate and control food safety hazards to prevent foodborne illnesses from happening. All right, air quality. Air is mostly made up of nitrogen, as you can see here, and 21% oxygen. And the sources of air pollution are mobile stations, stationary sources, air sources, and natural sources. And the exposure, of course, is via inhalation, obviously. The WHO states that there are now 6.7 million deaths a year worldwide due to ambient and household air pollution. And this picture up here on the right is actually a picture I took in June of 2023 in New York City when the wildfires in Canada were blowing over. And I was in Queens. I was leaving work and I was in Queens. And this was the picture that I took. And it was quite a time. I'd never seen anything like that. It was really an experience. When we talk about air pollutants, there are there are two major types of air pollutants the way we can think about it. One is primary air pollutants, and these are direct emissions. They come directly from the source. They can come directly from a stationary source or from a car. And indoor air pollutants are also examples of this, such as asbestos, radon, secondhand smoke, and volatile compounds as well. And this is compared to secondary air pollutants, which are created by chemical interactions in the air. Ozone is an example. We're going to talk about that next. Benzene and nitric oxides, and these can have quite an effect on health. Hazardous air pollutions, they are considered to be carcinogenic and can have other serious adverse effects. So things like benzene, perchloroethylene, which is found in dry cleaning substances, and methylene chloride, which is found, I think, in like industrial facilities or paint strippers as well. So these things are important to keep in mind. An act to remember is the Clean Air Act, which allows EPA to establish national ambient air quality standards, NAAQS, to set limits for the 6 air pollutants. And we're going to talk about that in the next step. I think it's also important to mention, I don't think I mentioned it later on the slides, but I want to mention it now. The kids are at a higher risk of getting toxicity from inhalation. And that's one of the reasons. There are many reasons why they're at risk for toxicity, but one of the reasons is they just breathe faster, so they take in more substance. Naturally, their rate of respiration is higher. NAAQS pollutants. You do have to know this, and you do need to know about the Clean Air Act. So it's important for your exam to know that. These are the 6 pollutants that have set limits by the EPA that need to be monitored. Sulfur oxide is a component of acid rain. And acid rain is just a broad term that includes any precipitation that's acidic. So it has like sulfuric acid or nitric acid that can fall down. And it's a high risk for people with those who are asthmatic, and as well as any susceptible conditions related to respiratory problems. 2, nitrogen oxide, which is a component of smog, which we're going to talk about in acid rain. Carbon monoxide, and this comes from unvented space heaters, leaking furnaces, exhausts, and it reduces oxygen to the body tissue. So the high risk of exposure to this are children and individuals with cardiovascular conditions. Ozone, which we're also going to talk about soon, causes airway irritation, coughing, and difficulty breathing, and again is high risk for those who have chronic respiratory conditions. Lead from air and ingestion, which can cause neurological effects. Again, high-risk children, kidney, immune development, and reproductive. It can affect those systems. And particulate matter. Particulate matter are very small pieces of matter that float in the air, and they're directly emitted into the air from factories, power plants, and cars. The thing with it is that it's so small that it can get pretty deep into your respiratory system, and sometimes if it's small enough, it can get through the alveoli and into the systemic system. So particulate matter is a really high concern, and of course high risk asthmatics as well. So I mentioned ozone. And ozone, there are two types of ozone to talk about. One is the good ozone. And you can see here, right over here, the good ozone is in the stratosphere right up here and absorbs most of the UVB radiation. Now remember, there are different types of UV, and UVB is the one that causes sunburns, right? So it's like sort of the Earth's sunscreen. It's protecting the Earth. It's good. And unfortunately, there are things such as ozone depleting substances that can get into the air and affect the ozone. But this is the good one. But then there's another kind of ozone. Sorry, this is the stratosphere ozone right here. And this is another ozone right here called surface level ozone, which is right over here, much lower. And this kind of ozone is a result of a chemical reaction between VOCs and NO and heat. So volatile compounds and NO come from industrial facilities, motor vehicle exhausts, gasoline vapors, and chemical solvents. So they're basically emissions that come from these places. And they combine together and they cause this sort of air right over here. And ozone is a big part of smog. And we'll talk about smog in the next slide, but smog is it's a combination of fog and smoke, right? That's how they come up with the word smog. And it's very, very toxic for your respiratory systems and as well as for your neurological systems. And as I mentioned, ozone is regulated by EPA. So important to know, two types of ozone, one is good and one is not so good. Here's a picture of smog. And the reasons why smog is harmful. It can, it can be irritating to your upper airways, and then in your lower airways can cause, asthma attacks, chest pain, and if, of course, you have a chronic respiratory condition, it can trigger that as well. And ozone, like I mentioned, ozone is a combination of oxygen combined with nitric oxide and heat. Smog is due to also not just ozone, but also the involvement of particulate matter, which we mentioned in the previous slide. It plays a big, big role in smog, both of those, ozone and PM, but also NO2 and sulfuric oxide as well. Sulfur dioxide, sorry, play a role in it too. But particulate matter and ozone are the major components of smog. This brings us to climate change. And the way we talk about climate change is it's a long-term shift in temperature and weather patterns. And some shifts can be natural, but they're usually pretty subtle. But when we have dramatic increases and dramatic changes, then we attribute it to climate change. For example, we know that the Earth's temperature has increased by 1.5 degrees Celsius in the last 100 years, which doesn't sound like a lot, but it is a lot. And even a few more degrees than that can have devastating effects on the world. And the reason this happens is because of something called greenhouse gases, which are released from human activities such as fossil fuel burning. And we can take a picture of greenhouse gases in a nutshell in this slide here. Burning fossil fuels releases greenhouse gases into the atmosphere. And the greenhouse gases are carbon dioxide, methane, nitrous oxide, fluorinated gases, and water vapor. And greenhouse gases, they absorb heat and they create this greenhouse effect. So they make the Earth a lot warmer than it generally should be, because usually some of that heat is dissipated out into space, but we're keeping it all within ourselves. This increases global temperatures and increases surface temperatures of the ocean, and we'll talk more about that in a second and the repercussions of that. So when the oceans warm up because of climate change, the glaciers melt, so the sea level goes up. The waters get warmer, so storms have more fuel because that warm air, the warm water really triggers hurricanes to be a lot more vicious, a lot more stronger, a lot bigger than they usually are. They can keep the warm seasons more prolonged. So things like mosquitoes are going to have a field day and they'll be doing their vector disease transmission for longer periods of time as well. Wildfires will be more rampant. They'll be harder to put out. And regular ecology will be affected. Natural habitats will be affected. Organisms will be affected across the board because of that. Also, there'll be higher CO2 levels, increased pollen, and that can increase asthma rates as well. So lots and lots of repercussions of climate change. And it's good to take a minute when you get a chance, so we don't have to go into this in detail, to think about how the impact of climate change can be in kind of a big picture perspective, as well as the little things we don't really think about and how they can be affected. So it's good to reflect on the massive consequences that can occur from climate change. Oh, I'm out of time. Okay, great. Okay, we'll switch gears and talk about water. There is a lot of water in the world, but only so much that we can really use. 95% of it is saline, and 2.5% only are freshwater, right? When we use our fresh water, precipitation sometimes can replenish our water supply, but sometimes it's still not fast enough. There are uses of desalination plants and things like that that are very useful, but they do come with a lot of energy, resources, and money. So they're not utilized quite so often. They are there, but it may not always be cost effective. So when we talk about water quality, it's influenced by two things, drought and flooding. Drought can lower the water table, and flooding can actually result in water pollution because, as you can imagine, there are lots of contaminants that are flowing into the water system. So those are two things to keep in mind. And when we think about water contamination, they can come from two places, or in two ways. One, it can be a point source. So we know exactly where this contamination is coming from. It is coming from this landfill. We can identify the exact location. And non-point source, that means we can't quite identify where it's coming from because it's coming from a different place. Like it's coming, a little bit's coming from a landfill, another part's coming from, septic systems. So that is usually from runoff. So those are two ways to think about the source of water contamination. Two important acts to know pertaining to water. One is the Clean Water Act, which established national standards and set limits on pollutant discharges. And the second one is the Safe Drinking Water Act, which allowed the EPA to set contaminant levels for water pollutants in drinking water. So over 90 agents. All right, very briefly, let's go through how we purify our drinking water. So our drinking water comes from two places, either surface water, which is usually like lakes, rivers, places like that, and groundwater, which is water from underground, obviously. The majority of our drinking water does come from groundwater. There's lots of stuff that needs to be removed from that, as you can imagine, from water coming from either sources. So the water is pumped towards a treatment plant. The first step of cleaning the water for drinking is to add chemicals called coagulants that have a positive charge on them. And what that does, it makes the particles that you want to remove much larger. And that makes it easier to get rid of. Then it goes into flocculation basins, which are basins that sort of gently mix the water. And when it mixes the water, it puts them together to form even larger particles called flocks. And in the next step, which is a sedimentation step, the water kind of floats around, and all those heavy particles that flock, it settles to the bottom. And all that stuff that settles to the bottom is then taken away to a landfill, and there's cleaner water on top. Afterwards, that water that is separated from the top and the bottom, the sludge from the bottom, all that clean water is taken over to the filtration tanks, and it goes through a filtration system, removing even more impurities. Finally, it goes into the last step, which is disinfection. It's usually a chemical disinfection. There are multiple ways to do disinfection, but chemical is the most common one, such as chlorine, and that kills any remaining organisms. The water is then stored in a clear well. It's a storage underground. Fluoride may be added, and then it's delivered to cities via pumps. So it's important to also know that drinking water that's sent out to the cities sometimes may contain trace amounts of chlorine and disinfectant, and that's okay. Please remember, drinking water is regulated by the EPA. They're in charge of it, and there are multiple types of public water systems that are listed here. There's community water system, non-transient community water systems, and transient non-community water systems. What is used is basically the community that it's supplying, as well as how often people consume the water in that community. And this on the left is sort of a brief understanding of how cleaning drinking water works, I mean. There are a couple waterborne pathogens that are also regulated by EPA. I've listed them here. Cryptosporidium is a parasite, and it is tolerant to chlorine. That's important to know, so it can be a little hard to get rid of. Giardia is another parasite that lives in the water also, and it's with contaminated feces. I never forget Giardia because it's associated with foul-smelling diarrhea. My parasitology professor in med school told us a story about how he had been around so many people with Giardia that he could identify someone had Giardia based upon their foul-smelling gas that they let out. And that just never left my head. And I always associate Giardia with foul-smelling diarrhea since then. It's a weird story. I never forgot it. Thought I'd share it. So hopefully you'll remember it too. In fact, Giardia is very interesting how it works in the body. This is just sort of an FYI I thought was interesting. It's the different cycles that Giardia takes inside your body when it gets in and just replicates and takes a new form. It's pretty interesting if you're curious. It's worth looking into if you're not familiar with it. Enteric viruses such as norovirus and rotavirus are also waterborne. And Legionella, we did talk about Legionella earlier. It becomes a problem when it infiltrates into human-made water systems and can cause respiratory illnesses, high fever, and headaches. Moving on to sewage. Sewage is proper disposal. Well, let me say this. Before our sewage system was created, people just dumped their sewage in the streets, or they used cesspools, or there was like an open sewage system. So obviously this opened a can of worms, literally and figuratively, as to the situation that we had, especially in regards to sanitary and sanitation and hygiene. And the first sewage systems in the US were in Chicago and Brooklyn in the 1850s. Because you watch these movies and you see people like dump their chamber pots on the street, like that actually did happen. So very fortunately, we have a fantastic system, a really, really interesting process as to how we deal with sewage. There are a couple ways to deal with sewage, actually. Rural areas and some of the more suburban areas sometimes can have septic tanks that collect wastewater, and then they use bacteria to break it down, and it needs to be pumped every 2 or 3 years. But obviously, this system wouldn't work in a major city like New York City, where I live. Municipalities need to use a different system. So this is a system that talks about what cities use. What we do is when we flush the toilet, the waste goes downwards underground and into a pipe, a really, really big pipe called the sewer main. So everybody in the area, their sewage all gets drained into the sewer main, and that all just gets fed into a wastewater plant. When it gets to a plant, it goes through this process that you see over here. And we can think about it as a primary treatment plant. First step is primary, then the secondary, and the tertiary. The primary, the first step, is more of a mechanical process. So basically, you're going through a screen, and there's a bar screen that's basically filtering out big products, big things that go into your, say you flush into your toilet, like toys or pans or things like that. The big stuff is filtered out that way. It then goes into a grit something called a grit chamber, which also further removes like sand and pebbles and other things that might have been missed that way. And then it goes into a settling tank when it's kind of rotated around a bit, and solids are separated, and floating scum goes to the top and sludge is on the bottom. The floating scum is usually stuff like grease or oil, and the stuff on the bottom is usually sludge. Both the scum and the sludge are both removed. And then it goes into this next section called the aeration tank. And this is really interesting because it's called an aeration tank because there are a lot of bubbles that are floating around that have oxygen in them. But what's fed into the system is something called activated sludge. So it's actually sludge that has bacteria in it, and it's fed into the system because that bacteria breaks down organic stuff, or organic material that we're trying to clean. So it breaks it down even more. After the aeration tank, it goes into the settling tank where stuff settles even more. It's also known as a clarifier. And some of the activated sludge from this stage is either fed back into aeration tank, or it's sent down into the sludge digester and then disposed of in that way. After it goes to the settling tank, it goes on into a disinfection tank where it's chlorinated and cleaned, and then it's sent out to rivers, lakes, and oceans. So the entire cycle takes about 24 to 36 hours. And the clean water that's released, it's called effluent release. Sometimes it goes into another treatment plant for additional cleaning. So pretty fascinating to know what happens after we flush a toilet. It's really interesting. There are some really great videos on YouTube about the cycle of sewage management. And I do recommend you looking at those animations because they do explain it in a very clear and a very interesting way. So here is another picture to just kind of break it down. It's laid out a little bit more clearly as to how sewage goes. The primary is mechanical. The secondary is more biological because we're using the activated sludge to break down organic products. And the tertiary is more disinfection. So. And a way that we know we can measure the effectiveness of a sewage system is by checking the BOD, the biochemical oxygen demand. And this is the amount of oxygen consumed by microorganisms while decomposing, right? And so the way I think about how to measure this is thinking about the word demand. If someone's demanding something, that's not good, right? So if there's a high demand, we're not happy because that means there's a lot of microorganisms in that water. So if there's a high demand, that means that there's a high rate of decomposing organics or organic material. We actually want the BOD to be lower. So we measure the effectiveness of the treatment because it should decrease with every step of the sewage treatment. The amount of BOD decreases 25 to 40%. It decreases even more at the next step, and then it decreases up to 99% in tertiary treatment. So a BOD is important to know to understand the effectiveness of the sewage treatment. And remember, active sludge or solids and liquids separated out of the wastewater. It's called sludge. All right. Solid waste management going on. Prior to the 1900s, solid waste was either thrown into the street, buried in the backyard, or even burned. But now, thankfully, in the mid-1900s, monitored landfills were created in our country. So thank goodness we have a way of getting rid of our trash without having to just dump it out. Landfills are usually, they take on whatever cannot be recycled or composted. So we want to try to recycle and compost as much as we can, and whatever we can't goes into a landfill. And municipalities are the ones responsible for disposal, either via landfills, recycling, or composting. This is how a landfill looks like. It's usually dug a little deeper than, it's dug deep into the ground. And there's usually a liner that's placed right over here. And there's a system that collects the liquid that may drain from the waste. So this area right over here is filled with solid waste, and then it's compressed all the way down. And with the compression, fluid kind of leaks out of it, of course. And that fluid is called leachants. So leachants are fluid that's drained through the waste and contains contaminants of the waste. And so there's a collection system that gets all of it over here. And it's very important to have the collection system because if we don't, it could seep into the groundwater and contaminate the groundwater. So that system is very, very important. Another important part of this is methane. So because there's organic compounds inside of the landfill, as they decompose, they can produce methane. And methane, as we know, is a greenhouse gas, and we need to control that. So there are actually federal regulations monitoring the leachate and the methane as well. The methane goes off and it's burnt to create electricity. And leachate is collected and sent off to a plant to be properly disposed of. So those are the two important things. There's also a cover that's usually put on top of a landfill, or should be put on top of a landfill to prevent odor as well as any sort of emissions from that. When the landfill stops accepting stuff, it's done. It's not accepting anymore. It'll be closed up, and it's monitored for many, many years after that for safety as well. That takes us to hazardous waste. Hazardous waste can have potentially serious health effects as well if it inappropriately disposed. We may have heard of Love Canal, which is an important event. But just to mention, hazardous waste can come from whole materials like pesticides, cleaning products, paint, car products, medical waste, industrial waste, radioactive waste, mining waste, lots of different places. And the way it's treated is various ways: chemically, thermally, biologically, physically, lots of ways to deal with hazard waste. But if you recall, the Love Canal tragedy there was a canal that was created kind of around Niagara Falls, and it was supposed to be a big project, but it got abandoned. And then a chemical company decided to dump a ton of chemicals in there. And then they sold it. It was 22,000, it was something crazy, like 22,000 tons of chemicals were dumped into the canal. It was just sealed off and then sold to the city. And the city built an entire community on it, like environment, the elementary schools, housing, things like that. But then the air started to smell. Children started coming home with chemical burns on them. And that was because the ground started to crack from the houses being built and all this and the built environment. And then people got very, very upset, obviously, and they rallied for a very, very long time because babies were being born with birth defects and miscarriages. People were getting sick. Leachates were migrating out of the water system, affecting ecosystems and the water supply. and a very high level of dioxin, which is a very scary toxin, very effective toxin, with very bad side effects were also found. Eventually, after many, many years, they finally cleaned up and evacuation occurred. But unfortunately, thousands and thousands of people were affected by this. And this just goes to show the importance of proper hazardous waste cleanup. As a result, the CERCLA/Superfund was created by the EPA. The CERCLA, also known as the Superfund, was an act that required responsible parties to assume liability for the cleanup of existing inactive and abandoned waste sites. And there are specific sites that are labeled Superfund sites. And the land, and this is usually lands contaminated by hazardous waste and identified by EPA as a candidate for cleanup. There's also a fund that was created, hence the word Superfund, that helps support cleanup in hazardous waste sites that have been unclaimed. The Resource Conservation and Recovery Act, RCRA, is very similar to CERCLA and gives EPA the authority to control hazardous waste at active sites. So it's similar to CERCLA, except CERCLA deals with the cleanup of existing inactive and abandoned sites, and RCRA deals with active sites, and it identifies hazardous waste based upon criteria. So these are two to know. All right. Here's another question. Okay, so here we go. Here we go. Hazardous waste. Here is a food safety system, HACCP, food safety system employed too. All right, good job, everyone. Identify and control problems that may cause foodborne illnesses before they happen. Awesome, very good. Detect bacterial contamination of foods after it happens. No, we're trying to prevent, right? HACCP is prevention strategies. So all this other stuff should have been done. Well, A and D should be done. Could be part of it, but the main purpose of it is this. So good job. All right, this takes us to our second to last section, which is toxicology and risk assessment. Toxicology is the study of adverse effects of chemicals. ADME is an abbreviation that's used in pharmacology and pharmacokinetics that stands for the cycle of the chemical in the body. The drug is absorbed, distributed, usually most commonly during the blood, but also in other ways as well, broken down and then excreted. It's a good cycle to remember because it helps you think about susceptible factors that people might have that can play a role in how that chemical affects their body. For example, individuals with kidney disease have to avoid drugs like NSAIDs because NSAIDs can be nephrotoxic. They can be toxic to the kidneys. Other things such as age play a role in the cycle as well. Like I mentioned, little children can be really susceptible for inhaling toxicity. But also they can be at risk because they're smaller, so a lower dose can affect their bodies more. They're still developing, so some of these toxicities can affect their development as well. I put in the number here for poison control. They're a very useful tool that we have nationally. So if there's any questions or concerns about poison consumption or exposure to poison, they're very, very helpful, and I've had to call them many times. So it's in my career, so very useful number to have. Again, several routes, ways that a chemical can be absorbed into the body. Which route the chemical chooses to take depends on the property and the pharmacokinetics of the drug. And just keep in mind, some of these chemicals can use multiple routes. Like, for example, lead can be ingested or it can be inhaled, right? And the most common route that leads to toxicity is inhalation. Other routes to take into consideration would be breast milk and transplacental absorption as well. Here's a list of common toxins. It is by no means exhaustive, but this is just to give you an idea of how we are exposed to toxins every day in ways that we don't even realize, how it's all around us all the time. We talked about a few of them, but some of them, we haven't really talked about, but hopefully some of them do look familiar to you. Asbestos is due to multiple reasons. The big one is wall insulation and associated with mesothelioma. And we're going to talk about that more in the next slide too. I also just want to mention there's something called PFAS that's come out. You might have heard about PFAS. And what PFAS is considered, quote unquote, a forever chemical because it never breaks down in the environment. And it's gotten a lot of traction recently in the news because it's something that's found in products like fluoropolymer coating or products that are heat resistant. So like food packaging, cooking utensils, and it can get into foods, it can get into animals. It's currently being studied. The effects are currently being studied. I'm seeing results and studies every day about the effects of PFAS. So the point of me mentioning that is that we're constantly learning about chemicals that can affect us. And we have to be on constant alert for things that we may just be recently learning about. So it's interesting to know and important to keep in mind about these things. So dose response curve. This helps us further understand how to consider risk when we're dealing with chemicals by looking at this curve. And in a nutshell, the dose response curve basically says that shows how differing levels of exposure can cause different effects on an organism, right? And these are all the key terms listed here that help you kind of understand the dose response curve. The lethal dose is the lowest dose that is fatal. And the LD50 is the dose that can cause 50% of the test population to die. And this is the most a measure of acute toxicity. Now, acute toxicity is defined as the adverse effect from one-time exposure. For example, if you ingest in a large amount of organophosphates, then you're going to have serious effects of toxicity pretty immediately, right? As opposed to chronic toxicity, which is adverse effects from long-term low-level exposure, like asbestos, like you've been exposed to asbestos for a long time, and then you're going to have symptoms later on in life. The idea is really the higher the dose, the higher the effect. And the one exception we have for this are carcinogens. There is no safe threshold for carcinogens. They are considered risky across at all times. There's nothing that we can say is a safe level. And a threshold dose is the minimal effective dose of any environmental agent that evokes a stated or all or none response. And again, carcinogens are the exception to this. By understanding this curve, we can start to figure out how to make decisions when it comes to the use of chemicals, and we can better understand what the risk is and compare it to the benefits so that we may get a desired outcome. For example, there's a drug called Coumadin, which is commonly used as an anticoagulant. However, it's necessary to check and monitor the levels of Coumadin to make sure that it's within a good range. If it's below therapeutic range, that means it's not beneficial enough and the dose needs to be changed. But if it's too high, then that's risky. And the patient will be at a higher risk for bleeding, such as with spontaneous bleeds. So that's just an example of why it's important to understand the relationship between dose and response. Which takes us to our next slide, which is understanding human health risk assessment. So understanding risk assessment is done using a 4-step risk assessment process. It's really the method of determining the probability of harm occurring to people from exposure to a hazard, right? So really all it's, in the simplest form, it's saying, let's gather all this information about our hazard and about our exposure, and let's take that information and determine what the risk is. So the first step is saying, identify the hazard. So what kind of adverse effects can be produced by the chemical? Second and third is dose response assessment. So examine the relationship between the dose and the effect of the dose. Of course, the dose response curve will be useful here. Also understand the exposure assessment. So understand the exposure itself, right? What is the potential exposure route, exposed population, how frequently the exposure occur, the magnitude, everything about the exposure, and take all that information we have learned. And we go to our next step, which is the risk characterization. So it's a quantitative, measurable, either quantitative or qualitative measure of risk associated with exposure. So it's basically an integration of the information gathered in the previous steps to determine and estimate the likelihood of an adverse effect occurring in an exposed population. Okay. And then this takes us to our last section. And this is just a very good summary to know about the laws associated and the policies and the acts that have come up over time. I know it can get a little tricky because some of these laws get amended and they have a new name, or there's an act that comes out of another act. So it can get a little tricky. I think it's good to focus on the big ones. I try to hit up a lot of the major ones during the presentation. I think we talked about Clean Air Act, Clean Water Act, Safe Drinking Water, CERCLA, RCRA, we talked about those. It's also good to know about the national, international agencies and laws that are available and what they do. So that will be useful to know also for your exam prep. So where am I with time? Oh, perfect. Okay, good. So that leaves 30 something minutes for questions. So I have practice questions. We'll go on to practice questions. Okay. All right, here we go.
Right,
Are
just
you
give me a
ready?
second. I'm trying to
Sure,
get
yeah.
the launched. All right. Everyone
Okay.
should see the pool now.
Okay, cool. The agency responsible for monitoring drinking water in the US. It's a pretty straightforward one.
We'll
So
give it about
do I click it?
15 more seconds.
Sorry.
Yeah, I'll close it and then I'll share it for
Okay.
you. So we'll leave it open for about 30, 45 seconds. This was a fairly short one, so I'm gonna leave it open for 30. Um, yep, we've already got like over 75%. So here we go. 96% said A.
Woohoo! Okay, good job, everyone. That is the correct answer, EPA. That's just straight memorization. There's no quick way to learn that one, just memorize it. So good job. All right. Next. 2-year-old child is found to have an elevated glucose lead level of 20. Upon inspection, it is found that the paint in his apartment contains high levels of lead, and the child has been eating the peeling paint. What step in the hazard assessment process does the inspection level represent?
Fifteen more seconds. Right. So this one, it looks like we have 59% of people said exposure assessment, and then we have 32% that said hazard identification, 8% said dose response assessment, and 1% said risk characterization.
Okay, great. So I would categorize this as exposure assessment. I know this can be a little tricky, but examining what the exposure was, right? Where was he exposed? What was the exposure? How long was he exposed to? So I would identify this more as exposure assessment. Um, I guess if we talked more about lead, you could say more about the hazard identification, but in this sense, I was looking more for exposure assessments. Okay, next question. Oh, here we go. Okay. The act that permits the EPA to hold responsible parties accountable for hazardous. Oh my gosh, I gave you the answer already. Oh no. Okay. Pretend you didn't see that. Okay. The act that permits the EPA to hold responsible parties accountable for hazardous waste and cleanup and also establishes a fund for hazardous waste cleanup is.
We'll see who was paying attention right
Oh
away.
no.
Give you about 10 more seconds. Okay, so for this one, it looks like we have 60% who said CERLA. We have 22% that said Toxic Substance Act Control, 10% said Clean Water, and then 8% Pollution Prevention Act.
Yeah, so CERCLA is the right answer. It holds responsible parties accountable for hazardous waste cleanup and also establishes a fund for hazardous waste cleanup. Now remember, CERCLA is also referred to as Superfund, right? So if you remember that, this question might be a little bit easier to figure out too. So CERCLA is the answer. Okay, I get this right this time. Oh, here we go. Okay, great. 65-year-old male is diagnosed with mesothelioma, a rare type of lung cancer. His occupational history includes many years of installing insulation during demolitions or renovations. The likely cause of the cancer is exposure to.
More seconds. All right, so this one, we had 99%.
Woo! Nice. Okay, awesome. Asbestos is the answer. Very good. Legionella obviously is not, there's nothing in the story that makes us think Legionella. Secondhand smoke, mercury, there's nothing here that makes us think that. So, okay, next question. The 65-year-old male diagnosed with mesothelioma is a rare type of lung cancer. His occupational history includes many years of installation. So the same guy. Most likely route of exposure to the causative factor is ingestion, inhalation, dermal entry, or injection.
Five more seconds. Okay. So once again, we have 97% that said inhalation.
Great. Good job. Inhalation is the number one way that asbestos is you can get exposure to asbestos. There is a small chance of dermal entry, but it's inhalation is by far the primary, the most common way.
And so since there's like both of them, I'd say for this one, I'd suggest the best answer. So the most common. I know that can be a little bit confusing, but the most common is the correct answer.
Okay, question 6. Leachate is contaminated liquid that percolates through solid waste disposal heights, viscous material that is a byproduct of wastewater treatment, large particles that are created during flocculation, overflow water from flooding.
Five more seconds. Okay, so this one we have 71% said A. And then we have 18% that said B, 8% said C, and 3% D.
Correct answer is A, contaminated liquid that percolates through solid waste disposal sites. That's the correct answer. B, viscous material that is a byproduct of wastewater treatment. That's more likely like sludge. Large particles that are created during flocculation, that is flock. And overflow of water from flooding, that's just something I made up. So A is the correct answer. Okay, moving on. Which of the following waterborne diseases organisms is very difficult to kill by chemical disinfection, by drinking and or recreational water supply systems, and has caused major U.S. disease outbreaks?
Seconds. Okay. So we have a little bit of a all over the place, if you can see
Yeah.
this one. It looks like 43% said B. 20A, 20 or 19C, 19D.
Okay, so the answer is cryptosporidium. It is a big problem because it's kind of hard to do a chemical disinfection of it. Cholera, which I think is the second most, the one that's voted. Cholera is actually very receptive to like chlorine treatment. It does really, really well with chlorine and disinfection. So cryptosporidium is the one that we are looking for here. Okay, moving on. In some cases of foodborne illnesses, hemolytic uremic syndrome is caused by which organism?
10 more seconds. So the majority said 65% said B.
And B is correct. Very good. This is just memorization again. E. coli 0157 is associated with severe diarrhea, like watery, bloody diarrhea. I can quite sick from it, and HUS is one of the things we are concerned about with this exposure. All right, question 9. What is the danger zone in which bacteria multiply in fast food enough to become a hazard?
For about 10 more seconds. Right. So 80% said C.
Very good. That is correct. 40 to 140 degrees is the danger zone that we need to be careful. Okay, question 10. Municipal sewage is typically processed to remove organic material before disposing the treated water. In large cities, this treatment generally consists of A, biological treatment in an aerated, activated sludge system, followed by settling to remove sludge solids, then chlorination prior to discharge. B, chemical treatment by coagulation and flocculation, followed by settling to remove solids, then chlorination prior to discharge. C, filtering through sandbeds to remove organic solids, followed by chlorination prior to discharge. And D, superchlorination to destroy organic materials, followed by discharge.
I'll leave this one open longer just because it's more reading. All right, we're going to close in 5 seconds. Okay, so B and A and B, it looks like we have really close. So we got 46% said A and 48 said B.
Yeah, this is a little bit of a tricky one. The answer is A. We really want to think about the process in which the sewage water goes through. It is a little tricky. I too would be between A and B. But if you look at the cycle of the sewage, water flowing through sewage, you could see the first step is the biological treatment. The second step is biological treatment, and then then it's removed, the sludge is removed, and then it's chlorination prior to discharge. Chemical treatment, coagulation, flocculation is more of a mechanical treatment than a chemical treatment. So that's the big one here, followed by acetylene to remove solids, and then chlorination prior to discharge. So just remember, coagulation and flocculation is primary treatment, which is mechanical. Secondary treatment, which is what's explained in A, is biological treatment. And then the third step is chemical treatment. So I think that's really probably what it was. But this is a little bit of a tougher question. So good job. Okay, question 11. Sewage treatment can be considered effective if the biological oxygen demand A increases, B decreases, remains unchanged, or fluctuates.
Ten more seconds on this one. Okay, so this one, 73% said decreases.
And that is right. Decreases, man, demand. If somebody is demanding things, it's not a good thing. That's how I think about it, right? If that means the bacteria that's there that we're trying to get rid of is demanding more, and we want that demand to go down. That's how I think of it. Because sometimes it can be a little confusing. So good job. Next question. Which of the following is not considered a greenhouse gas?
Wait, 5 more seconds. Okay, so we have 52% said water vapor and 37% said oxygen.
So
So
the
our
answer,
two ones.
oxygen is the answer. The other four are considered greenhouse gases. Yep. So that's just need to know it, right? We breathe in oxygen, so it shouldn't be a greenhouse gas. But the other ones are. So, okay. In addition to ozone and particulates, which of the following pollutants are most likely to aggravate asthma?
Great, 10 more seconds. All right, this one, there's two of them. 47% said sulfur dioxide, and then we've got 37% that said carbon monoxide.
The answer is sulfur dioxide. So carbon monoxide, remember, tends to affect the cardiovascular system more. Carbon monoxide poisoning, it affects it affects the heart more. That's the way to think about it. I do want to go back because I think I had a typo in slide 12. I put carbon monoxide, but greenhouse gas is carbon dioxide. So I left the 2 out after the O. So the A should be carbon dioxide. Carbon dioxide is a greenhouse gas. So those of you who pick CO, you are not wrong. You are correct. I made a typo in that slide. So, okay. Yes, so sulfur dioxide is the answer. Okay, moving on. Question 14. While visiting a woodland area in North Carolina, a 40-year-old woman developed fever and muscle pain. A few days later, she also developed a flat, light pink, splotchy, red, non-itchy rash which started on her hands and feet. The causative agent and vector that caused this disease are. There are a couple ways to approach this question, so I'm curious to see how everybody does.
I'm gonna give everyone 10 more seconds. Okay, so we have 69% that said D.
Yes, very, very good. Okay, so the answer, this is Rocky Mountain spotted fever, and it's due to Rickettsia rickettsii, which is the vector is a tick. The other, so let's talk through this, right? So Yersinia pestis is the flea. That's the one that's associated with the plague. Generally, this is how it presents. You have large lymph nodes. You can have like bleeding into your skin and things like that. The rash is very suggestive of rickettsia. But also, you can look at the choices like Borrelia-Burgdorferi, which is due to Lyme disease. And we know Lyme disease is not transmitted via a mosquito. So you can rule out B just because Borrelia-Burgdorferi and mosquito aren't together. And West Nile virus does not present this way either. It doesn't is usually associated with a rash. So it's a little bit of a tricky one, but I just kind of wanted you to think about how you can rule things out when you look at the answers. Okay, which of the following best describes how vaccines work? Now I'll let you read that. It's a lot of words.
10 more seconds. So 83% said A.
And A is correct. Well done. Most vaccines work by triggering the body's adaptive immune system, including antibody production that can target a specific pathogen. Vaccines don't provide antibodies. We have to create our antibodies. Most vaccines trigger the body's innate immune system, which results in macrophages. No, it's not innate, it's adaptive. And most vaccines work through epigenetic mechanisms that turn on genes to produce specific antibodies. That's not correct. Next question, characteristics, attributes, or exposures such as hypertension, unsafe sex, alcohol consumption, unsafe water that can increase the likelihood of developing disease or injury are known as A, risk factors, B, environmental factors, C, causal factors, or D, community factors.
Okay, 15 more seconds. So 86% said A.
Well done, risk factors. Yep, that's exactly what it is. This is examples of risk factors. Which of the following is currently the most common cause of unintentional deaths in the US?
I learned this today from you.
Yeah.
10 more seconds. Okay. So we have 64% that said B and 28% said A.
Yes, unintentional poisoning is correct. Usually due to overdoses, again, because of the increased use of opioids. Motor vehicle accidents used to be the most common cause and has since changed. Moving on, head lice can be transferred to another individual through sharing hats. In this scenario, hats are examples of being. So this one's a tricky one. Think about what you think would be the best answer, because it could potentially be another one.
Okay. 5 more seconds. Okay, so we have a bit of a mix, but 50%, or sorry, 56% said C. That was our largest one.
So the answer I was looking for was fomites. So a fomite is a type of vehicle. And so it could technically, vehicle could be right, but fomite is a more specific way of defining what that vehicle was. Because a vehicle could be anything. It could be air, it could be water, or it could be a fomite. So it's technically not wrong, vehicle, but specifically I'm looking for fomites. So fomites are inanimate. They're just inanimate objects, right? So the hats are inanimate. So vehicles, you're not wrong saying vehicles, but fomites are a little bit more specific. So, but I did want to put this question here to demonstrate that fomites are a type of vehicle. And yeah, so, but you're not wrong if you said vehicle, you're not wrong. So, all right, going on. Which of these groups of conditions include one or more single gene disorders that are part of the recommended uniform newborn screening panel?
About 10 more seconds. Okay, this one, 73%, all of the above.
Well done, that is correct. We test for all of this in a newborn screen. And finally, a health clinic is required to report all cases of chlamydia to the local health department. This is an example of the clinic sending information to the health department. Active surveillance, passive surveillance, syndrome surveillance, or sentinel surveillance.
Right, 10 more seconds. Okay. All right, so this one, we had 46% said A and 31% said B.
Okay, so the answer is B, because the health clinic is sending out, and there's a law that requires them to send it out, but they're sending it out themselves. The health department is not requesting that information being sent to them. So it's being passively sent over to the health department. So if the question was the health department is required, I guess if you say the health department, it reaches out to the health clinic to send information regarding this, then you could say it's active. I guess I could see how you might think it was active, but what I was trying to say, it's required on their own to report all cases of chlamydia. So, but if I said the local health department has reached out to the health department and requesting to send that information out, then it would be active. I hope that makes sense. Maybe I could have worded that better, but good job. That is it. I hope you found this useful. I think there are a couple questions in the Q&A, right? I can pull them up.
Yep.
So, okay. Someone wants to post the defects for the different chromosomes again. I don't think you need to know the numbers. Someone's saying can someone post the defects for the different chromosomes again, please? I can type that to you. But you don't need to know chromosome numbers. But let me type this just for an FYI. Cystic fibrosis is chromosome 7. PKU, I think, is 12. Yeah, and then sickle cell disease is 11. Okay, but you don't need to know chromosome numbers. That's just an example I was giving. Okay, what is the difference between smog and greenhouses? So smog is the air that's created with a combination of ozone and particulate matter that kind of sits on the bottom. It doesn't, ozone itself, it can be a greenhouse gas alone, but smog is a combination of other elements. So it's not considered a greenhouse, but it has an element of a greenhouse. I hope that makes sense. Could you please repeat the types of exposures that cause lamb's defects in babies? Martina, could you clarify that? Causes lamb's defects in babies, which sorry, if you could clarify that, because I'm not sure what lamb's defects you mean by that. Okay, where do gunshot wounds fall on the cause of death currently? So let's look back. If you go to the CDC, they actually lay this out pretty well. Let me pull up that chart and we can look at to see where that is. Where are we here with this? Okay, we look at this chart over here. Gunshot wounds. I guess it depends if it's suicide or if it's homicide, right? And where would it line? It probably in all other unintentional. So probably in this gray area over here where they kind of lumped it together. I don't have that exact statistic, but I know the CDC does. So I'd recommend that you go check the CDC site for the exact number as to where it lies and if it's number 5 or number 4, because I'm not sure. What is sentinel surveillance? Again, sentinel surveillance is when you are surveying a specific group of healthcare facilities. So you're kind of like gearing it towards a small group as opposed to like everybody. You're not reaching out to everybody. So you're able to really hone in and get more detailed information at specific illnesses because you're focusing on a group. I'm waiting for whoever asked that question on the defects to tell me what they meant, but I think it was Martina who had asked about the defects. If you could throw it in the chat because I wasn't quite sure what you meant. But if not, I guess they can email.
Yeah,
Oh. Okay. Is there any law that prevents mothers from, there's 2 more questions. Mothers from smoking during pregnancy? Unfortunately not. There's no law that says that you can't. And limbs. Limbs. Gotcha. Limbs defect. Oh, thank you. The limbs defect. I think you were talking about the thalidomide, right? We were discussing thalidomide, and when mothers are taking thalidomide in the 1950s and the 60s. I'm trying to find that slide. Here we go. When they took thalidomide in the 1950s and 60s, the infants were unfortunately born with these limb defects. Their limbs were like flippers and other sort of defects as well. So anyways, I think that's all the questions.
Yeah, I think so too. Okay, so as far as next, our last session is next week or is on Thursday. So I hope that you will all attend. It's on law and ethics. And I want to thank you for being here today and especially to our presenter. So thank you so much for being here and we will see you on Thursday.
Thank you, everybody. Bye.
Bye.